Hello everyone, my name is Walid Faisal. I'm a project manager and career fit fellow at the School of Pharmacy in University College Cork in Ireland. And today I will talk with you about the microneedle technology as a smart horizon to deliver medications. So the hypodermic needles, it's very old technology used for drug administrations. Many shortcomings, starting from the needle phobia because of the pain caused of touching the nerve endings. We have a waste of the needles, which may cause needle sticks injury. And we, of course, heard about the cold chain transportation challenge, with, especially with vaccination in the last year with the COVID. Okay. So to tackle these shortcomings, we innovated the microneedle-based drug delivery systems, which is a microstructure needle, which is long enough to penetrate the skin, especially the stratum corneum, to deliver the medication, and it's short enough so it will not touch the nerve endings and will not cause the pain. This is enable an alternative delivery scenario for people, and will, because it has no pain, it will improve the adherence to drug regimen and will reduce the burden on health systems and increasing the coverage of drug administrations. So what's a microneedle? The microneedle itself is a microstructure, sharp needles in the size of microns, and this one, when it's applied to the skin, will penetrate the stratum corneum of the skin, start to dissolve, and release the medication locally. One array patch of 1 centimeter squares can carry up till hundreds of microneedles. So the microneedles itself has different designs, from an old solid design to the hollow microneedles, the coated microneedles, and the dissolvable microneedles. The dissolvable microneedles is the most technology used of microneedles, where you mix the drug and the polymer in a suitable solvent. After dissolving them, you put the solvent in a hollow mold and leave them to dry, and then you collect your needle with adhesive substrate. Then when you apply it to the skin, it penetrates the stratum corneum, and then the polymer starts to dissolve and releasing the medication inside the skin to give the effect of the microneedles. So, however, there is a lot of challenge for this technology. The first challenge is the drug loading. As we know, the size of the microneedle is very small, so you are limited with the amount of the drug, but then you need the microneedle to have this structure of sharp microneedles. It's mostly formed by the excipient, the polymer itself. And the polymer has to be around 90% or more to give this strong and sharp structure of microneedles, and this limiting the amount of the drug loading. Again, presence of the polymer and the excipient has many issues with the regulatory, as to have to confirm you need a clinical study for long-term use of topical microneedles. Of course, the cost of the production is so high, especially when we work with a new technology of productions which has no infrastructure. So we identify an opportunity to develop a new microneedle technology. We call it array patch, which is able to penetrate the skin barrier and deliver the medication locally. And also it's produced, the unique character of it, it's produced from 100% of medication, whereas there is no need of polymer in this formulation. And this is a convenient for the patient, for the application, and also you decrease the cost of production by using a simple method of production, and also there is no need of regulatory clinical trials for the polymers. So how we produce it? It's simply, it's a technology where you have the melting the medication at high temperature, and then when you have the melt, you fill your mold with the melted medication, cool it down, and then you collect your needles with adhesive substrate. This technology is, to make it simple, is something similar to production of the glass sheets or glass bottle by heating sand. This technology was patented and published last year in 2020, and we claim in this that we have 100% drug loading and using the novel melt process of manufacturing. This technology is a platform technology where we can use it when any suitable meltable drugs, which has a physical-chemical character suitable for melting. And there is a lot of molecules under this category, like the antifungals, anti-cancer medications, corticosteroids, and anti-inflammatory medications. When you go for the design of microneedles, we are successfully produce a sharp microneedles which enable to penetrate the skin and deliver the medication locally. And as you can see from this sharp microstructure needles. Also, we try this in the using the big skin model for the penetration, and we can see from the picture that it's the efficacy of the microneedle to penetrate the skin and deliver the medication intradermally. We look for the feedback from the stakeholders and the key opinion leaders in the industrial stakeholder confirm there is unmet need in the market for this microneedle technology, and they also confirm that the best pathway to start with is the use for the microneedle in the treatment of the nail fungal infections. Also, the clinicians were very interested in the idea of localized effective wave treatment of onychomycosis, and especially because of the high side effects with the current treatment. So why they choose the nail fungal infections? The nail fungal infection or onychomycosis has a high prevalence, or 10% of the population is chronic, resistant to treatment. 16 to 25 patients will never achieve the full treatments, and they have a high recurrence rate. The market of nail fungal infection is 4.6 billion in 2017, and we have a growth rate of 6.7 in the next forecast of 2018 to 2026. The reason of this is the overpopulations, especially in geriatric patients. When you talk about the site of application, the main site of application is the nail fold, which is skin layer, which composed of the nail root and the nail matrix. And this is the place where the fungi is resided. So when you go to the chemical and physical stability, the confirmed by the drug content, texture analyzer, and imaging, and we also use XRD and DSC and LC mass and HPLC and NMR to confirm the stability. We use to confirm the efficacy, we use the minimum inhibition concentration technique, and we confirm the suitability of gamma radiation to sterilize the microneedles. So when we look for the diffractogram, the XRD diffractogram and the DSC curves, we can see that the melting of the itraconazole and we can see the glassy structure of the glassy amorphous structure of the melt, which is stable over 3 months as amorphous structure. The NMR also confirms that there is no difference observed between the itraconazole and its melt over 3 months, which indicating the chemical stability. The HPLC data confirms there is no degradation peak, and all what we observe is less than 2% of degradation, which confirms the chemical stability. LC mass analysis confirms the high stability of itraconazole before and after melt, with having same fragmentation pattern. When we use the minimum inhibition concentration, again, it's different fungi and yeast strains, which is implicated in onychomycosis. The data in the table confirms that the melt and has the same efficacy as the ABI, and also the melt after 3 months still giving the same efficacy. When you talk about the work progress, we can see we are progressing now the in vivo technical development of itraconazole microneedles, and we also identify and validating the best market to follow, and we're seeking potential manufacturing and clinical partner for collaboration in this technology. Finally, I would like to thank the group working with me, Dr. Ziad Sertawi and Akash, Professor Abayna Kreen and Fatima Farag for the technical advising, and the commercial group of Claire Wallace, Kevin Dalton, and Nicole Martinelli. And thank you, and